Thank you very much. And they were great words, weren't there, about there being a lot of opportunity. So change is scary, but it's also exciting. And I guess when I was thinking about all those three speakers and what they were talking about, a lot of the times they were thinking like customers, thinking like patients, thinking like people who use the blue book and then put it somewhere and have no idea where it is, and all that information can't be accessed. So think like your patients, think like doctors, think like nurses, and ask, what can we do that'll make life easier for them, more seamless for them, and that'll guide you, I would suggest, to places that are open and ripe for innovation. Now, the last of these sessions, there will be one more keynote after this and a couple of quick other things, but the last of these three-headed sessions is about disruptive technologies changing the way we treat and care for patients. So disruptive technologies have already helped solve many healthcare problems. Thousands now have access to their genetic data so they can see if they've got a predisposition to certain conditions. Wearable devices let us monitor vital signs. Surgical robots help surgeons perform tasks they could never ever previously have imagined performing. What disruptive technologies are next, and how will they change the way patients are treated and cared for? So to discuss that again, three speakers: Marcus Dorr, founder and CEO of Health Horizon, followed by Professor David Thomas, Director of the Kinghorn Cancer Centre at the Garvan Institute of Medical Research, and then Professor John Rasko, AO, Director and Head of Department at the Department of Cell and Molecular Therapies at RPA. So firstly, please welcome Marcus Dorr.
Thanks, James. Hi, my name is Marcus Dorr, and I'm a data scientist, a CEO and an entrepreneur, if you can't tell from the skivvy. This conference has been fantastic in that we've seen a whole lot of innovations that are coming out of medtech, biotech, e-health. And the thing is that we're not alone here. Every day around the world, there are conferences like this with engaged people like yourselves, looking at innovations, trying to engage with innovations. Now, what I really wanted to do today in this forum is to tell you about a way that I can put you at the centre of all this innovation that's going on. You'll basically go away today and say, well, I saw some great innovations, and you'll talk about them, you'll tell people about them, and then you'll move on next week, next month, and you'll say, oh, that innovation, where's it gone? Where's it going? And I've got a way to show you how you can engage with that. And indeed, this is happening globally. Now, why are you here? And it's really that you are an engaged health consumer, a practitioner, you're involved in the care of others. And there's a society emerging around the world of people like us. And it's very heartening to see the transformation of health. And what my group is really trying to do is put you at that centre. So these are all the innovations that are coming out of New South Wales. Our group at Health Horizon has been working and looking at all the different kinds of innovations coming out of the groups. You might see one of the groups here that you're involved with, or that you know about, or you've heard here at this conference. It's really, really important that there's innovations coming right from hospitals, right through to clinics, and also the whole innovation ecosystem that's started up and is going on in Australia and around the world, where clinical innovators but also entrepreneurs are teaming up, and you see this kind of whole ecosystem emerging. And it's really critical, because this is really the passion, the energy that's pushing through, and not just being the usual way in which medical innovation has been done previously. This is really a revolution. So basically, 10 years ago, I started in health as a data scientist, but also as a company director, and looking at the innovations coming out of hospitals, and particularly around New South Wales and ACT. Now, I got to about 500 innovations that we built in a small spreadsheet that we had coming out of the hospitals in New South Wales, and I looked at it and I thought, well, how can we help these innovations on? They need funding, they need collaborations, and there was a real gap in that whole area. And really what we looked at then was, well, can we make a place that we can put all these innovations so that people can find them in a meaningful way? Now, 10 years later, and right now, our group has catalogued over 500,000 data points around innovations around the world as a major contribution to the world's health ecosystem. And in Australia, we're doing this right down to city level, right down to the individual institutes level. Now, when you're trying to wrangle all of that data, all the innovations that are happening and the different stages and things like that, what is actually meaningful? What's meaningful for you? And we brought it down to these really three concepts, which is, what is it? What is the innovation? What does it do? What is the actual improvement, or what does the innovation achieve? What's its impact? And then also, what stage is it at? The thing is that innovation is a spectrum. It starts probably down in research, and then as it gets translated, it goes through trials and the like. The pathways to innovation are not all standard. And we're seeing that with digital health, for example, where digital health apps, for example, they don't start with the usual evidence-based approach necessarily, but they gather evidence over use. As long as they don't do harm, then the efficacy is proven in real-world data. And that's what regulators are currently all grappling with at the moment. So we use this basic taxonomy, a way of classifying all the innovations that we've found around the world, and we've put them up for people to be able to find and follow. So if we look at some of the innovations, New South Wales has got a great medical devices focus and fund and training program. It's bringing out and servicing some great innovations out of our hospitals and different research groups, and really trying to make them meaningful. So what we've done at Health Horizon is basically put these innovations up, and we're able to track them. The concept of evidence is really critical to our approach. As we know, in medicine, in health, the application of real-world evidence and clinical research evidence is really, really important for showing harm and efficacy, minimising harm and maximising efficacy. So we validate the innovations that are coming onto our platform that we find with a curated process of actual clinicians, and also some very smart AI algorithms that look at things that are like other things, and then they promote these up to our basically our clinical team to say, well, this looks like a bona fide and evidence-based innovation, let's put that up. Now, because we've got over 500,000 of these in our database, we don't have them all curated. That's a long process. So we're identifying different innovations that have a public interest, that people are calling for, rare diseases, for example, pharma failures, but also devices and also apps. Because within the app environment, we need more validation around things like reviews of apps by groups, by clinically valid groups. So innovations do cover the whole spectrum, and we found everything from the medtech, biotech, e-health systems that you may have heard about here. You haven't really heard about much public health campaigns, for example. They're pretty critical for us in terms of a spectrum of innovations, where you've got public health campaigns that worked, eradicating chlamydia in university environments, for example. They should be shared across, not just in journals, but actually more widely for groups to be able to get onto. It's all about promoting the innovation across the whole spectrum. So what we do is we also follow all the progresses of all the innovations that we've got on there. So you can look at these innovations and say, oh, they've been through these stages. This is the stage it's at. And so you can go and follow that innovation. So any of you can log on with your email address, look at the innovations you like, you can follow them, and then you'll be informed about their progress and where they're at. And this is really democratising the information in health. So we're doing New South Wales. We're trying to complete our New South Wales picture. We're doing a whole lot of scanning around Australia, and just yesterday we were awarded an MTP Connect grant, a Commonwealth Government grant, to complete our picture of the Australian health innovation system. So my request to all of you is, if you see a good innovation, you can nominate that innovation. it'll go into our curation process to basically service it. These are all innovations that are publicly available, that are ready to disclose. It's not back in research land that has IP that's not protected or anything like that. These are ones that are on the path to translation that could have meaning for you in terms of different disease groups, different medical fields, and we're very, very excited by the catalogue that we've built. We're seed funded by 2 non-for-profits. I'm the founder, co-founder, I've got as well, and we basically put our initial funds in, and we've now got 20 wealthy investors around the world who believe that this is a great thing for the world, and we're providing this as a free service as long as we can. So I recommend you get on there and you find and follow the innovations that are of interest to you. So we've already mapped a whole lot of innovation sectors in health in Canada. We've just finished Tel Aviv and looking at Israel. We're focusing on Germany. Australia is our big focus with our latest grant to have it'll be the first country that's got a complete scan of the innovations that are happening in health and medical technology and innovation generally. So this is really about looking what's on the horizon. Your health choices in the future may actually be determined by what's coming, not what's right available now. And in fact, engaging with innovations means something could go to crowdfunding, for example, and that happens often in failures of pharma, where you get a rare disease, you get some evidence for 2 drugs in combination that work on a particular disease, but both those pharmaceutical companies aren't interested in doing any more. So it can go to crowdfunding, you suddenly get a crowdfunded study, and then it's on its way. Those things are happening all the time. Funding innovation is really, really important, and you're at the centre of that. So you can basically now be kept up to date by following those innovations, and then you are empowered. And our idea and our challenge with Health Horizon is to empower everybody to know what's coming in health, to know things that are bona fide, so it's a validated set of innovations, and you can follow that. Now, on top of that, we have the ability to accumulate and group those innovations together in channels, and we've created a channel for the health innovations in New South Wales, ones you've seen here yesterday and today, and you can go and find those innovations you've heard about, and you can follow them, and I certainly recommend you go and do that. And thanks very much.
Well, it's a great pleasure to be able to talk to you today about where I hope that research, and particularly genomics and imaging, might have an impact upon cancer in our community. I've never been in a forum where the ideas have been so edgy and dangerous we've required an ambulance before. So how do you make an impact on cancer? And the themes that I want to talk about today are that we don't have to be brilliant here. Like most ideas that are important, we stand on the shoulders of giants. And looking at the impact of science and research on clinical outcomes from heart disease is an easy way to solve a very difficult problem. What is the problem? Well, as you see here, there will be over 130,000 cancers diagnosed roughly this year. 47,500+ people will die from that. And of those, 1 in 3 deaths will be premature. And that premature mortality from cancer should really worry us, because it amounts in aggregate to 88,000 years of life lost to disease every year, and $4.2 billion of lost in foregone annual income. The lessons here are that the earlier you get diagnosed with a disease, the greater the impact and loss to our community that follows. So how can technologies like genomics potentially impact on all of this? Well, let's look at cardiovascular disease. This has to be one of the most spectacular graphs I've ever stood in front of. It shows the reductions since 1985 in mortality due to cardiovascular disease in our community. That's more than a 60% reduction. This impact on cardiovascular deaths in our community has substantially contributed to the overall improvements in lifespan. Now, what could this be due to? Cardiovascular disease, like cancer, has many fathers, so to speak. It has hereditary causes, it has environmental causes, and there's a certain random element to this as well. What about smoking? Well, cancer hasn't done as well, for a start, and you see that here over the past period of time, same period of time, 30-odd years, you see there's been about a 10 to 20% reduction in mortality. If it's smoking-related that's improved cardiovascular disease, you'd expect lung cancer to have improved, and so that's not the answer here. Although smoking cessation has been enormous in terms of its impact upon cancer and cardiovascular disease, it's not the answer to this improvement in survival in cardiovascular disease. What about screening and early detection? Well, I'm a medical oncologist. I treat patients, and most of the patients I treat have advanced disease by the time they get to me. So I don't often get a chance to look after people when there's a chance to cure disease. But in breast cancer space, where we've introduced public health screening, breast cancer has outperformed other cancer types and particularly outperformed lung cancer. So the trick here is to try and treat the patient when the disease is localized on the right, as you can see it, rather than expending money and resources when effectively the die is cast. In this situation to the left, barring the exception of immunotherapies potentially now, it's not clear that any drug treatments that we have will cure this patient. But on the right, good old-fashioned surgery has a very good chance of curing this person's lung cancer. So how do we pick up people, particularly young people with cancer, before it gets too late? Well, the lessons from cardiovascular disease are that you need to treat the risk factor and not the disease. And in cardiovascular disease, we identified risk factors like hyperlipidemia, like high blood pressure, like diabetes. And by modifying those risk factors before we had to deal with a stroke or the myocardial infarction, we managed to improve. That's, I would argue, the major contributor to that improvement in survival. That and a concept which we haven't introduced into cancer, which is about secondary prevention. Once we have a diagnosis of a myocardial infarction, it's routine in the cardiovascular world to focus upon that patient to minimize subsequent outcomes. So, for cancer, what is the risk factor that we now have available to us? Well, cancer is a genetic disease, and genomic technologies essentially allow us to understand potentially everything about our risk factors for disease. And particularly risk factors like genetics are important in early onset diseases. So, out of the 6 billion nucleotides at 3 billion positions in every human's genome, they specify everything from our height to our eye color to our risk for developing cancer and cardiovascular disease. And with technologies like the HiSeq-X, the Southern Hemisphere's largest genomics facility now housed in New South Wales, for the first time we're able to ask questions about what we'll learn about the genetic risk factor for cancer and how we can use that to try and alleviate the mortality, not just alter the survival of patients with advanced disease where cure is not realistically possible, but how do we actually save lives? So this is imaging, believe it or not, and it shows the impact of a companion technology, which is very important. It's about whole-body magnetic resonance imaging. This, for the first time, allows us to move beyond screening for breast cancer and bowel cancer, which is where we focus most of our preventive strategies, that and cervical cancer, to asking questions about cancers that could arise anywhere in the body. And here you see an example of a patient of ours that we enrolled onto a program by virtue of genetically identified risk, who had a cancer located in their flank muscles completely silently, and this tumor, which had a high-grade element indicated by the arrow, was removed curatively as a consequence of therapy. This woman was 50 years of age. It's not just one woman, though. Science needs to advance by numbers. And this shows a paper that we put together worldwide as part of a consortium, and showing that the use of whole-body MRI in individuals at very high genetic risk of cancer identifies cancers in between 1 in 14 and 1 in 7 individuals on a single scan. Now, these technologies are not cheap, so the trick here is to use the technology of genomics to identify those at risk so we can get the cost-benefit ratio as high as we possibly can. But already in this very high risk, we see the promise of combining these two different technologies in patient populations at risk. So our vision is that by expanding out into the young onset cancer population, a subset of the entire community who've already declared themselves to be likely carriers of genetic markers of risk, we might improve survival in that group. So how many young people are there diagnosed? Is this a significant problem? There are more than 6,000 individuals who are diagnosed with cancer under the age of 40 in Australia. Our cure rates in this group are pleasingly, not pleasingly enough, but 85%. And that means there are over 200,000 individuals who are survivors of cancer under the age of 40 in whom we could potentially intervene by secondary prevention strategies that we might learn from cardiovascular disease. And this group has an extremely high post-treatment morbidity and mortality. The best data that we have come from the Children's Cancer Survivorship study, and they show the data following treatment and curative treatment over a 30-year period. So just think, when we're talking about a 30-year period, that sounds like a long period of time. But if a person is diagnosed and treated at the age of 5, that's age 35 by the end of this graph. If they have a cancer diagnosed at 15, that's 50 years of age. And after the risk of recurrence and progression from their primary cancer, the risk of secondary malignancies rises and rises until it becomes the major likely cause of death in this population afterwards. What I'm proposing as my risky idea that might transform cancer mortality and improve cancer control in our community is to take every survivor of cancer which arises under the age of 40 and screen them to try and identify that subgroup which is at the greatest risk. Our current estimates are that using this technology, about 1 in 4 people who are screened will carry a marker that suggests they are going to be in that group that develops second malignancies. It's not just about discovery, because when you move from research in animal systems, you can discard the experiment once you've made your discovery. But when you've got a patient in front of you, there's a necessity to try and close the loop and use what we can from imaging and other technologies to try and offer something for these patients. And we can demonstrate that in the highest risk group we can make a difference. And what I see in the future is that we'll take this information and we'll apply it to those 6,000 people who get cancer every year under the age of 40, and then use that information to invest in ways of curing them and ensuring that they have long, healthy, productive lives. Thank you.
Good afternoon, everybody. This is an exciting moment for me. I would first of all like to acknowledge the traditional owners of the land on which we meet and their elders past and present. I am a physician, scientist and haematologist who has the pleasure and indeed the honour of leading 2 departments, the Cell and Molecular Therapies Department at Royal Prince Alfred Hospital and another department of Gene and Stem Cell Therapy at the University of Sydney Centenary Institute, as well as an organisation that is the International Society for Cellular Therapy, which I'll become president of next year. All of those organisations humble me and remind me to acknowledge the fact that I'm a public servant. And I serve my masters political and direct, and I owe a great debt to them personally for encouraging me and helping me to achieve the goal that I'll be talking about for the next 10 minutes. Namely, the delivery of genes as medicine. I am the last of the 3 great disruptors today, and I want to try and convince you that there's something new, not just on the horizon, but here and now. I'm also mindful just to make one distracting comment at the start, which is that I was so taken with the presentations earlier of the jaw reconstruction, especially by Kai Cheng and Jonathan Clark, that I found myself literally tearing up in the audience as I saw that Asian lady's face being reconstructed. And I'm furious at Payal for making me cry in the audience before a talk. It is no mistake that I'm speaking after a fantastic friend and colleague who is one of the preeminent deliverers of genomic medicine. And what I want to try and convince you is that there is a future beyond genomic medicine and genetics, which is gene therapy and cell therapy. I'm saying that genetics is so last century that you probably even recognise these, grey photographs of gentlemen who are from the middle of the last century. Gregor Mendel on one side, the famous pea priest, and on the other side, of course, every scientist geek's favourite person to worship, Charles Darwin with natural selection. And, Gregor Mendel even then said, genetics is so 1860s, to which Charles Darwin, of course, would have replied, "You got that right, bro." That's a James O'Loughlin joke because, bro, he was a priest. And okay. I'm reminded of the common saying when we have our offspring that you have to play the hand you're dealt. In work every day, you have the resources that you use and you play the hand that you're dealt. It's a common saying, it's one that we're all familiar with. And indeed, it extends to the fact that we may be very clever because our parents are very clever. We may be great athletes. We may have specific skills in life. But it's not just the cards that you're dealt. And I want you to think about it in an entirely different way. When you consider it's not just the cards, perhaps the economic benefits, but it's the genes that determine so much of our lives. Cleverness, prettiness, athletic prowess is to a great extent determined in that way. But as you can see in this slide, it's also genetic diseases hidden in the 3 billion bases, as you've just heard, that determine our predisposition to various different diseases, some being caused by single disease genes like haemophilia, cystic fibrosis, other diseases caused by multiple abnormalities in genes like cancer, diabetes, Alzheimer's. Nonetheless, the idea that one day we may be able to correct the root cause of disease is what has driven me to work and skip to work for 25 years as we've been developing these technologies. And it's my aim today to convince you that they're here and now. So what is it that I'm talking about? Genes are chemicals, strings of C's, A's, G's and T's that is encapsulated in our DNA in all of our cells. They cannot work outside of a cellular context. So gene therapy is always married with cellular therapy, and that's why I wear another hat with stem cells and cellular therapies, because genes, whether they are used to introduce genetic material and information into a cell in the body, whether it be injected into a vein and then finds its way to a cell, or whether it's injected directly into a tumour, or whether cells are removed from the body, in both cases it's a living therapy. And so here's the paradigm shift. Here's the disruptive idea. You only need to give this therapy once if it works okay, and then that should be the cure. Because the idea here is you're dealt a bad gene, a bad card. Why can't we use that information to then put back the normal version of that gene? That's the idea that compels me. It's so straightforward. It's been very technically difficult to achieve because the difficulty has been in getting an efficient, sufficient amount of genes in to get them to transform the cells. But in the next 3 examples, I want to at least give you some insight into how we are currently, today, achieving clinically meaningful results. Example number 1. Only in the last 6-8 weeks have we heard that the United States Food and Drug Administration has approved 2 new cell gene therapies, immuno-cell gene therapies for leukemia in childhood and young adults, and in lymphoma for adults. The latter of the trials we were involved in at Royal Prince Alfred Hospital, one of only 2 centres in Australia. This idea is called chimeric antigen receptor T cells, and it basically uses one's own body's immune cells reprogrammed outside of the body and then used to target specific markers on leukemias and lymphomas that are unique and kills those cells. In a sense, one always asks oneself a question when you're faced by someone who's suffering from cancer and has to break that horrible news. Why isn't the body's own immune cells attacking the cancer and ridding it? It's because the cancers often have molecular means of putting the immune cells to sleep. And this reprogramming technology activates these cells and makes them specifically target, like a cruise missile, diving in on a particular cancerous target and killing those cells. The response rates in highly pre-treated and resistant patients has been nothing less than astounding. 85% response rates in patients with leukemia. And if you don't believe me, believe the market. When the announcement was made only 6 weeks ago of the 2 companies, one of them was subsequently purchased a couple of days around that date for $12 billion US. So, how do we do it? You've got cancer, leukemia or lymphoma. Your own cells are removed from your body and then shipped to a manufacturing centre like the one at Royal Prince Alfred Hospital, of which there's only a couple or a few in Australia qualified to be able to do this for clinical manufacture. Those cells are then genetically modified using a virus so that they now target specifically markers on the surface of the cancer cells. Those cells are then expanded in the laboratory in a sterile and safe manner, and then they are returned to the bedside where they are infused with chemotherapy into that same patient. So one's own cells being genetically reprogrammed and put back into the body. This is the first image of these cells, a scanning electron micrograph that Bruce Levine shared with me. These are the genetically reprogrammed human immune cells that are now wrapping themselves around these nanoballs that have been coated with the specific marker called CD19 that now allow them to multiply in the body. They multiply thousands and, in fact, tens, millions of fold. And these cells have been called serial killers because each one of these reprogrammed immune cells can kill not just 1,000, but tens of thousands of different cancer cells. Remember, it's a living therapy. So it's not like a drug where you need to give it again and again, multiple cycles, multiple treatments of radiotherapy. It's a single injection, your own cells, they grow and multiply like your immune system, and then they kill the target cells. And when they're finished, they return back to the normal state where they are monitoring the body for the presence of any relapse. Well, seeing is believing, and here is the picture of the actual results of that kind of therapy. 5-6 years ago now, this little dog was suffering. This little girl was suffering from severe, relapsed, multiply resistant acute lymphoblastic leukemia. And at the University of Philadelphia, led by Carl June, Bruce Levine, Stefan Grupp, and their colleagues, they infused this little girl, Emily Whitehead, with one of the first CAR T-cell therapies. She's now 6 years cancer-free, a single injection. That's one of the approvals that have been granted in the last 6 weeks in the United States. The cost, $475,000, which has been justified because the company has now said we're only going to seek reimbursement if the patient responds. An entirely new way of seeing health economics with implications to the whole way that we deliver health, and we anticipate this will expand to all of these different types of technologies. Emily Whitehead has become one of the loudest proponents with her family that has spoken to represent the interests of developing this technology and the results that I've just described. Example number 2. This is a therapy that Jean Bennett has developed for blindness, a disease called Leber's congenital amaurosis. It's a blinding condition that causes kids to become completely blind within their early life. A single injection that Jean Bennett has developed over the last 20 years at the back of the eye, an infinitesimally small amount of volume of virus carrying the protein that they lack, leads to 13 out of 20 being restored vision and none of the controls. And the design of the trial was extremely clever because only one eye was injected with the gene therapy. And so it meant that patients were their own controls. This little boy is legally blind. He's doing a clinically approved maze, a test, and he can't even see the stop sign in front of him because the eye that was treated has been pasted over. He's only seeing through his blind eye and not seeing anything. He can't make any progress and isn't brave enough to go through the hurdles or follow the arrows on the ground through the maze. He received a single injection at the back of his eye of the gene therapy, and then within a month, changes were being seen, and these are sustained way beyond a year so far in the groups. And as you can see, he can navigate the maze, he can pass over hurdles, he can see where he's going and follow the arrows. He now rides a bike, catches balls, and can read where in the eye that was treated, and subsequently has received treatment in the second eye, so that he now has binocular vision. A compelling example which we hope to bring to New South Wales in the coming months and years. The final example is one that I'm especially passionate about because it's been the example that I've worked on for 20 years, which is the treatment for the bleeding disorder haemophilia, which you know was spread throughout the royal houses of Europe, probably the first genetic disease ever that humanity recognised by virtue of the fact that male children were bleeding during circumcision, and it was noted by the Talmudic and rabbinic scholars that that was something that could be passed through the maternal lineage, probably one of the first, if not the first, genetic disease. So Haemophilia B is a bleeding disorder wherein patients need to inject themselves 2-3 times a week with factor concentrates. In this case, it's factor 9, and We have, for 20 years, been pursuing a gene therapy to overcome the technical immune challenges, which meant that now our clinical trial to be presented at the American Society of Haematology in about a month's time shows 10 out of 10 patients being cured. And I think this is probably the only data slide that you'll see in terms of graphical data this morning and this afternoon. But for me, each one of these coloured lines show that a treatment that has converted patients who have severe haemophilia at less than 1% of the normal level, that's what severe haemophilia is, to reach levels above 12 and 15%, which is essentially means that they will never have spontaneous bleeds or never need to inject themselves again. None of these patients have had to inject themselves again with factor concentrate, with follow-up beyond a year in all of them. And each one of those lines represents a human being who is injecting themselves 2 or 3 times a week at a cost of between $100,000 and $250,000 per annum to the taxpayer. A single injection. Think about the implications economic. But one last point that I want to make, and this is the tragedy of haemophilia, is that 2/3 of individuals on the planet have no access to factor concentrate. It's not available because it's expensive and their country can't afford it. One day we imagine the possibility of being able to ship individuals with this disease and any one of the 4,000 other genetic diseases that afflict humankind with this kind of treatment so that we may be able to cure them and then send them on their way to have a happy life. There have been a number of technical challenges that have been overcome, those including gene transfer efficiency, the fact that safety is preeminently important and we are compelled to make certain that at no time do we cause any problems, and this has really been a major issue, as well as understanding the importance of regulatory compliance and strict attention to detail. As the chair of the Gene Technology Technical Advisory Committee for the Gene Technology Regulator of the federal government, I hold that position very responsibly, and I'm very, very well aware of the public concerns about the possibilities of playing God and the risks to the environment and operators. But every single disease has its own specific form of gene delivery necessary and an understanding of the particular genes that will benefit those individuals. In the not too distant future, we imagine the possibility of not just pasting genes in, but actually using so-called gene editing technologies to, in a sense, use molecular scissors to come in and actually correct the genetic abnormality present in the sequence of the DNA. And so one day your surgeon may not look like this kind of surgeon that Pale looks like, or many of the surgeons that you've seen speak today. They may look a little bit more, well, no, not like that, but molecular scissors anyway. You get the idea. Every one of these technologies that I've described require a particular laboratory to undertake this. And they are laboratories that, for example, have air 1,000-fold more pure than the air we're currently breathing. They have environmental monitors, strict compliance requirements, and everything is done by standard operating procedures. But above all, in addition to the infrastructure costs, which are substantial, that New South Wales is investing in, and I hope will continue to expand its investment in, it's an investment in people who are hospital scientists, clinical trials coordinators, and medical scientists, as well as physicians, all working together to try and achieve the goal of gene medicine. So I want to say that gene therapy is probably already out of the bottle. The gene has come out of the bottle, and I've shown you some examples today that suggest that although we may not immediately be here in terms of here and now being able to prescribe a gene, the clinical trial data in so many different diseases, and in addition diseases like thalassemia, where we've done clinical trials, immune deficiencies, some of the first successes shown, all of these suggest that the future is very, very bright for gene therapy and gene and cell therapy together. So in conclusion, I want to argue with you that gene therapy and cell therapy is definitely a gene that's now out of the bottle.
Well done. Yeah, that was good.